Hello everyone, Kitty Clinical here with the next part of the Biochem series on Step 1 Thoughts. We're going to be talking about mutations in the DNA. So there's different types of mutations. The degree of change, silent, is like not that big a deal. Missense, a little bigger a deal. Nonsense, a bigger deal. And frame shift, huge deal, right? So single nucleotide substitutions are repaired by both the DNA polymerase and ligase. There's types of single nucleotide point mutations are transition, which goes from purine to purine, and transversion, which goes from like purine to pyrimidine. So transition can be purine to purine or pyrimidine to pyrimidine. Transversion goes from purine to pyrimidine or pyrimidine to purine. And may I remind you again that CG is the CG bond has 3 H bonds, which means it's a little stronger than the AT bond, right? So that might come into play when it comes to these mutations. Moving on to single nucleotide substitutions, you've got the silent mutation, which is where the mutation codes for the same amino acid. Usually the third position of the codon, because remember we have that wobble effect that we talked about in our previous video. The missense mutation results in changed amino acids, so that it's also like, you'll see this in like sickle cell disease where you substitute glutamic acid with valine. Nonsense mutation results in early stop codon. Remember, the stop codons are UGA, UAA, or UAG. Usually generates a non-functional protein. And then there's other mutations too, like the frame shift is where you delete an entire deletion or insertion of any number of nucleotides not divisible by 3, which causes a misreading of all the nucleotides downstream, because remember that they're reading it in 3s, right? So if you screw up one, then it's like everything is off cycle, so it all goes south. Proteins may be shorter or longer, and its function may be disrupted or altered. Certain examples of the frame shift mutation includes Duchenne muscular dystrophy. Tay-Sachs disease can also be one. Splice site mutation is where you retain an intron in the mRNA, so the protein has an impaired or altered function. Some examples of this are some cancers, dementia, epilepsy, some types of beta thalassemias, Gaucher disease, Marfan syndrome. All right. So if you don't know about the differences between silent, missense, nonsense, frame shift, insertion, deletion, I would take the time to maybe go on Google Images and look it up yourself, and it would behoove you to kind of understand that a little bit. I also want to talk about the lac operon. This is a very classic example of a genetic response to an environmental change. It's probably something you've remembered in biology class, honestly. We talk about the lac operon gene. Glucose is the preferred metabolic substrate in E. coli, but when glucose is absent and lactose is available, then the lac operon is activated to switch to lactose metabolism. So the mechanism of switch goes like this. You have low glucose. That leads to an increase in adenylate cyclase activity, which leads to an increase in generation of cyclic AMP from ATP, which activates the catabolic activator protein, or CAP, which leads to an increase in transcription. When you have high lactose, that can lead to the unbinding of the repressor proteins from repressor or operator site, which also leads to increased transcription. So it would also behoove you to look up the different diagrams on the lac operon gene. I'm not going to go through all of it, but I can briefly mention that glucose inhibits adenylate cyclase, which brings ATP to cyclic AMP. So ATP to CAMP via the adenylate cyclase, which is inhibited by glucose. CAMP goes to it goes to CAP, binds CAP site, induces transcription, goes to the CAP site, and then that activates the promoter. You've got the operator next to it. And then you've got various like LAC-Z, LAC-Y, LAC-A. So the CAP site all the way, plus the promoter, operator, LAC-Z, LAC-Y, LAC-A is the lac operon. And a little bit further away from that, before the CAP site is the lacal, I think it's lacal, yeah. Repressor protein, which binds the operator, blocks transcription. it has it has allolactose, which is an inducer, and inactivated repressors when it combines. You have to look at the diagram to really understand what I'm talking about, but it is something that is important to know. You've got the various genes we talked about, LAC-Z, going to the messenger RNA to AUG, the state at which these LAC genes are strongly expressed or not expressed. So let's talk about that really quickly. When you have low glucose and lactose available, then the LAC gene is going to be strongly expressed because you're going to have the cap with the RNA polymerase together. When you have high glucose and lactose unavailable, that's when you get the repressor protein. So the LAC gene is not going to be expressed. When you have low glucose and lactose unavailable, then you're going to have the LAC gene not expressed either. And when you have high glucose or lactose available, then you have very low or basal expression. So you can try to memorize the states in which LAC gene will be expressed or repressed or whatever it's called, expressed, not expressed. It's going to be very complex. It's going to be somewhat not very high yield for the boards or for the licensing exam. I would say that I've maybe had like one question on the LAC operon in all of like the USMLE. And if they do give you a LAC operon question, they'll probably do it in a way that it's like a study, like, oh, this study tested was trying to evaluate the promoter gene of the LAC operon. I don't know, just they'll come up with something, right? But LAC operon doesn't really show up that much. What's probably more useful is honestly the mutations in the DNA. I would say that's going to be pretty high yield, knowing the difference between missense, frame shift, all that stuff. But take the time to study this. Hope you learned something today. I will see you in the next one.